So this is a molecular representation of a nuclear localization signal. So this is the part of the dengue polymerase protein that binds to our import receptors to take it into the nucleus of our cells, and that's how it's able to go in and shut down our immune response. And here we've got the phenylalanine, so that's the ring structure, which was really convenient to have a nice beautiful stone set into, which you guys have taken advantage of in the structure to make it look really cool. And the cool thing is it's the only flavivirus to have this C-terminal nuclear localization signal as well. So there's only dengue 2 and dengue 3 that have this, and no other one does, which is kind of cool. And that's our little alpha helice as well. So the protein that this binds to is called importin alpha, and it's called that because it's made up of 30 of these alpha helices. And this is representative of one of these alpha helices from which these will bind to. So this binds to about 12 of them, and the other 30 are just free in the molecule. So these go hand in hand because they're the binding partners. These are what come together in the molecule to make that stable. So this, the phenylalanine, is quite a large molecule. As we can see, it's quite broad to the others, and that enables it to get into the nice hydrophobic pockets and to really bind and help with the presentation of the signal. So that the key residues are these KRFR residues. So this sits within what's called the P4 binding site within importin alpha, so it binds a pocket. So the importin alpha has these helices, and between two helices is a pocket where parts of the molecule can go in to bind. So this phenylalanine goes and binds within it, while we've got this one sitting in another pocket, this one sitting in a different pocket again. So each different part of these four is sitting within a different pocket of importin alpha. So if this wasn't if the phenylalanine wasn't the right molecule, it wouldn't have bound in properly. So thankfully we've got these short parts of the molecule solved with it, but what it could hopefully mean is if we can find out what it means for the other types of dengue a bit more, we can start directing more specific targets to potentially just this region. Like if the nuclear localization, which this signal mediates, is critical for the virus, we can then target peptides to come in and hopefully bind to this to stop it getting onto our importin alpha and into the cells. So it may be a potential drug target. Like we have inhibitors that stop nuclear transport and act against dengue already, that's shown to kill all four types, but we don't know the mechanism of action per se. We don't know where it binds to, but if we already know how they interface, it'll be easier to then make a target against this interaction.
So did you understand any of that, Shannon?
No.